All right, it's after 1, so we'll get started. Thanks everybody for joining us. I'm going to kind of uncomfortably talk next to you, Kyle, so maybe people can hear me. Thanks everybody for joining us either in person or virtually today for Kyle's defense. Kyle came to us almost exactly 2 years ago to work on this project in Central South Dakota, looking at walleye and smallmouth bass interactions out there. So we got to spend a summer out in Central South Dakota collecting lots and lots of fish to look at what they're puking up. But I always like to throw out some fun numbers. I think for Kyle's, it ended up being about 3,500 adult diets that he looked at. So lots of good, fun goo and vertebrae and Callithra ID to figure out what fish are eating. So I'm not going to take too much time away from him, but I guess the other thing I was going to mention is exciting news for Kyle is that just last week he accepted a position with Florida. And so he's going to be departing here in just a couple of weeks. So hopefully today goes well for him so he can move on and carry on to his next position. So congratulations to Kyle. And with that, I'll let him take it over.
Awesome. Thank you for that introduction. Before I get started, I just want to give some acknowledgments. First, I want to acknowledge my funding sources, which is the Department of Natural Resource Ecology and Management here at Iowa State University, as well as the South Dakota Game Fish and Parks. I'd also like to thank the GFP biologists, their technicians, and our technicians here at ISU for help in the field and the lab work. I'd like to thank my committee, Dr. Weber, Dr. Morris, and Dr. Wanamaker. And then lastly, special thanks to Dr. Finsel and Dr. Grouse-Grubber for their input and contribution to this research. So today I'm going to be talking about interactions between walleye and smallmouth bass in Missouri River reservoirs. And to begin, we're kind of going to give a short background. Oh, it wants to switch. Okay. So we're going to start off with walleye in general. They're a popular sport fish, but unfortunately, they've been experiencing population declines for factors such as unsuitable spawning habitat, limited natural reproduction, and high angler harvests. So as a result, throughout the Midwest, several supplemental stocking initiatives have been put in place to help supplement declining populations. However, whenever you stock fish, there's always concern regarding predation and survival of your stocked individuals. So looking at the literature for post-stocking predation of stocked walleye, we found that it could be highly variable mortality-wise, anywhere from 0 to 100%, which is as variable as it gets. So therefore, you should look at it more from a species-specific standpoint. And some of the top predators linked to post-stocking predation of stocked walleye include adult walleye, largemouth bass, northern pike, and smallmouth bass. However, there's more concerns regarding expansion of bass populations and their effects on post-stocking predation of walleye. So looking at the literature specifically for that, there's a lot on largemouth bass predation on stocked walleye. In Wisconsin, they found post-stocking predation was associated with increases in largemouth bass populations. In Illinois, 4.3% of stocked walleye consume largemouth bass. And in Iowa, they found 11% of largemouth bass diets were composed of stocked walleye. However, comparatively, looking at smallmouth bass, there isn't literature on smallmouth bass predation on stocked walleye specifically. However, there is 2 studies in South Dakota that found that walleye made up less than 5% of smallmouth bass diets. However, these were naturally occurring walleye. And so that lack in literature would be important to help aid management decisions and systems where they're planning to stock walleye that have smallmouth bass to understand what those interactions are. And so the systems we're going to be focusing on today are in that situation. So we're going to be talking about the Missouri River reservoirs. They are renowned walleye fisheries. So people come from all over to fish them for their walleye fisheries. So it's economically important to the areas surrounding. Therefore, when walleye populations began to decline, supplemental stocking initiatives were set in place to help with that. However, there are smallmouth bass within this system. They were not native to the system. They were stocked in the 80s for a novel angling experience. And so now there's potential concerns with walleye declining, whether angler and biologists believe that smallmouth bass may be the cause for these walleye declines, either by predating directly on the stocked walleye or competing with adult walleye. And so to kind of assess these concerns, we need to look at kind of a food web and feeding ecology study to look at what the two are eating, the prey, and whether if things become limited, if potential competition will occur. So kind of looking at the food web configuration of the Missouri River reservoirs, the top prey fishes within these systems are rainbow smelt, cisco, and gizzard shad. However, their populations can be very cyclical, meaning they're very boom or bust. They can be abundant in one year and not in the other. And therefore, if they are competing for these or overlapping with these resources, if they become limited, then there's a potential for competition. And so one study looked at Lake Sharp, which is one of the Missouri River reservoirs, and they found that there was no adverse effects between smallmouth bass and walleye when overlap was high and consisted primarily of a large proportion of gizzard shad within their diet. However, when gizzard shad became limited, the two partition prey resources, however, both experienced declines in condition and growth. Therefore, furthering the knowledge out there to understand interactions between the two species and the presence of varying prey abundance would be important. And so the way to kind of address a study like this, there's two methods to look at kind of a feeding ecology approach. The first would be stomach content analysis, where you could get a collection of diets. And so you can see exactly what they're eating at a given time by IDing down to the lowest taxonomic level if possible. And so the good thing with this is you can see exactly what they're eating, but the bad part is it's kind of the short term of what they're eating. And seasonally, diets can change. And so a way to see, and another thing is it's very labor intensive to do this. So if you wanted to see a broader period of time, you would have to continuously sample. However, a way to see kind of the long-term diet integration is incorporating stable isotope analysis. And so with this, you're able to essentially see long-term the average of kind of what they're eating, because you are what you eat, less excrete. And so your isotope signatures reflect the resources you're acquiring energy from. And so some other things that you can see with this is where they occupy an isotopic space from a niche standpoint, or the role an organism plays in the community, as well as the trophic position, or where the organism sits on the food chain. And so for this study, we decided to combine the two to ultimately address our two concerns. And so my second chapter of my thesis and my first research chapter is to evaluate smallmouth bass predation on stocked age-0 walleye. And the third chapter and my second research chapter is to assess potential competition between smallmouth bass and walleye. And so we'll start with just focusing on that first chapter. And so the study site for this first chapter is Lake Oahe. It is a very large system. It's one of the largest reservoirs in America by volume. It starts in Pierre, South Dakota, and extends all the way out to Bismarck, North Dakota. However, because of how large the system is, logistically, we couldn't sample the entirety of Lake Oahe. So for this study, we focused on lower Lake Oahe, where there's 3 bays. We have Okobojo Bay, Cow Creek, and Spring Creek. And so in lower Lake Oahe, walleye populations have been declining, and there hasn't been natural reproduction in quite some time. And so there's been supplemental stocking initiatives set in place. And so we decided to use those bays and stocking to make a study design for this. So in June 2019, we had Spring Creek and Okobojo Bay stocked, and Cow Creek acted as a reference site. Now we were going to sample in 2020, but due to COVID-19, we were not able to. So instead, we ended up sampling in June 2021. And so for this year, we had Spring and Cow Creek be the stocked bays, whereas Okobojo Bay acted as a reference site. So I kind of want to focus on the reference sites. As I mentioned before, there hasn't been naturally reoccurring walleye. However, we took 2 measures to ensure that. And so before stocking, we did pre-stocking sampling throughout all 3 bays, on which we collected diets to make sure there was no walleye in there, because that would imply that there was natural reproduction. And then our second measure was having a reference site where post-stocking, we continued to sample that one the same duration to ensure that there was no walleye within the system as well. And then to kind of limit bias of a specific site, we switched to a different site in the second year. And so to collect all of our data, we use boat electrofishing, where we got total length in millimeters and weight in grams of all predators. But we fitted them with individual jaw tags, and we would use a fin clip so that we can recognize a recapture if the tag was removed or fell off. And then we use something called post-gastric lavage, in which you insert a tube down the esophagus of a fish. It squirts water down there and causes them to regurgitate their stomach contents, in which we collected them. And we bring them back to the lab to ID down to the lowest taxonomic level. And then we would get wet weight in grams of all of them so that we can have percent composition by weight. Now, when we had a highly digested individual, a way that helped us a lot in IDing was using Clathrum IDs. And so with that, I'll get into my first specific question for this chapter, which is what is the frequency of occurrence of age-old walleye in small bass diets? And so we got this from a very simple proportion. It's just the number of small bass with walleye in their stomach above the number of small bass with stomach contents. And so here on the top panel, we have Okoboji Bay in 2019, and the bottom panel is Spring Creek in 2019. Our x-axis is the small bass total length in millimeters, and then our y is frequency of occurrence. And so there's going to be two color bars that I'll show in a second. The gray ones will be ones that had stomach contents but no walleye, and the black bar will be ones that had at least one age-old walleye. I just want to make it very clear, though, that this is whether they had it or not, and not the number of walleye within their stomach. And so before I get into the results, I just wanted to mention for pre-stocking, as I mentioned, we did that. No walleye were found in the diets in both years, so all of the data is going to be from post-stocking. And so post-stocking in 2019 occurred from June 18th to September 17th. And for Okoboji Bay in 2019, we found that 4.9% of small bass consumed at least one age-old walleye. And those individuals that consumed them range from 285 to 391 millimeters. Whereas for Spring Creek, it had a higher percentage at 11.4%, and individuals that consumed them were from 209 to 416. And in Cow Creek, our reference site, no age-old walleye were consumed, which was good because, as I mentioned, this was kind of our measure to ensure that the ones that we were collecting were, in fact, potentially the ones that we stocked. And the size range that captured was from 192 to 464, which fit the size ranges that were shown in the previous bays to consume. So it's not like we were missing any. So moving on to 2021, post-stocking predation occurred from June 11th to September 28th. And for Cow Creek, it was 2.9%, and those individuals ranged from 252 to 362. Whereas for Spring Creek, it was also kind of a lower percentage at 1.6% and ranged from 276 to 290. And then for Okoboji Bay, there was no age-old walleye consumed. So comparatively to 2019, we saw lower percentages in 2021. So moving on to my next specific objective, I wanted to know when and how long is post-stocking predation a concern. And so we did this using mean proportion by weight. And so once again, the panels are set up the same way as before. However, the x-axis now is going to be days post-stocking, and the y is the mean proportion by weight of age-old walleye in the stomach. And so the highest percentage of age-old walleye within the stomachs occurred on 3 days post-stocking. It was at 5.8% of the diets, and predation lasted for 15 days. Whereas for Spring Creek, it was high as 1 day post-stocking, but that percentage was much higher at 38.6%. And it lasted for a longer period of time, for 25 days. However, it's worth noting that 2 walleye were found in the diets collected in September. However, that was months later, but still worth noting. So moving on to 2021 for Cow Creek, it was highest on 3 days post-stocking, but we saw our largest percentage within a single day at 43.2%, whereas it lasted predation only for 2 days. And then for Spring Creek, it was highest on 2 days post-stocking. However, the percentage was once again kind of low on the 8.3% and lasted for 3 days. So comparatively to 2019, predation lasted a lot longer in 2019, whereas predation only lasted up to 3 days in 2021. And so my last objective for this specific chapter is looking at the percentage of stock walleye consumed by smallmouth bass. So we did this by taking two things and adding them together. And so the first aspect was using our mark recapture data and to estimate our schnabel population estimates. And then we paired that with our package of Fish Biogenics 4.0, which is a biogenic modeling that takes into account water temperature that we gathered from hobo-loggers that were set out in the field, percent composition by weight of prey items that we calculated in the lab, predator and prey energy densities that we got from the literature, and then predator mean initial and final weights. And so essentially what this package does is it gives you the average grams a single smallmouth bass consumed of age-old walleye in a day. And so then we would multiply that by our population estimate to get a total number of grams of age-old walleye consumed by the smallmouth bass population. Now we want to get from the number of grams to how many walleye are estimated to consume. And so we used our age-old walleye growth estimate and divided by that to essentially get a total number of walleye that smallmouth bass consumed per day. And then we can look at that temporally for each day, and then throughout time, put it all together for an estimate to see a total consumed. And so the panels are still set up how they have been with the other slides. And however, this time Y is going to be the cumulative percent of age-old walleye consumed. So it's going to continuously get higher as more is consumed. And so our solid line will be the mean, and the dash is going to be 95% confidence intervals. And so for 2019 in Okobojo Bay, we found 0.6% of the walleye were consumed, or about 1,000 of 183,000 stocked. Whereas for Spring Creek, it was a higher percentage at 5.7%, roughly around 10,000 of 181,000 stocked. Whereas looking at 2021 for Cow Creek, it was at 1.3%, or almost 5,000 of 358,000 stocked. Whereas for Spring Creek in 2021, it was a lower percentage at only 0.2%, or roughly 600 of 257,000 stocked. So now I'm going to give just kind of a brief predation summary / discussion about my specific objectives. And so starting with frequency of occurrence, in a single bay, we saw up to 11.4% of smallmouth diets were composed of age-old walleye. However, looking at our overall mean for a year, we saw up to 5.9%, which was similar to other locations that represented both naturally occurring and stocked walleye. So compared to the literature, we didn't see much difference. Whereas for the size ranges that consumed them, we found 209 to 416 millimeters were the individuals that were known to consume stocked walleye. And then looking at the timing and duration standpoint, our highest proportion of age-old walleye and smallmouth bass diets occurred within 3 days after stocking. However, compared to the literature, it said that peak mortality generally occurred within 1 to 2 days. So we saw that it lasted kind of a day longer than what was previously gone for other predation studies on stocked walleye. And then for smallmouth bass, we saw that age-old walleye lasted in their diets for up to 25 days, whereas the majority of predation in the literature lasted 14 days. So again, we saw kind of predation last a little longer than what the literature stated. Now on to kind of the final one, which was the most important one, how many walleye are being consumed. And so we estimated that small bass consumed up to 5.7% of stocked age-old walleye. However, as I mentioned in the introduction, there isn't literature looking at specifically small bass predation on stocked walleye, but there is a study that saw that they consume up to 23.4% of saugai, which is a sauger and walleye hybrid. And then comparatively, looking at largemouth bass predation on stocked walleye, it was found to consume up to 100% of stocked walleye. So based on those, our observation of a small bass consuming just 5.7% appears to be negligible compared to the literature. So now I'm going to leave off this chapter with just some management recommendations. The first one might surprise everyone. The first thing is to do nothing. When you stock fish, ultimately, there is going to be some level of predation, and 6% comparatively to other studies is not a lot. So potentially you could be okay with that. However, if you wanted to find ways to help increase survival potentially, looking at bass behaviors may help you get to that. So what I have here is our population estimates for 2019. And we can see the black dot is June 18th to June 29th, and the white is July 1st to July 14th. And we can see that as time goes on, the population, the abundance of small bass decreases. And so what we noticed was they would be near shore, but as the summer goes on and the water heats up, they'll go deeper where it's cooler. And so potentially waiting to delay stocking to a period of time when there's less abundance of small bass may, as a result, end up with better survival. However, if you can't delay stocking, potentially sampling your bays for small bass density and stocking one that has lower density could, again, also potentially help with survival. And then the last one depends on logistics if possible, but if you're able to stock roughly 6% more to get your kind of desired survival rate, keeping in mind that potentially up to 6% can be due to losses, excuse me, then that's a way that you can go as well. And so with that, I'll now move on to my third chapter where I assess potential competition between small bass and walleye. Excuse me. And so a big part of this study is looking at changes in interactions between small bass and walleye in systems with varying prey assemblages. And so the first system we're going to be looking at is Lake Oahe. It is a very deep and large system that can support cold water prey fishing such as cisco and rainbow smelt, which are the primary prey species within here. However, historically, there's been a cyclical abundance of rainbow smelt. There's been a collapse around kind of the early 2001, 2002 range that occurred in Lake Oahe. And so the prey assemblage can be kind of cyclical within this system compared to Lake Sharp, which is a shallower warm water body of water that has gizzard shad as their primary prey fish that's pretty abundant throughout the year. And so to collect diet collection, we did it the same as the previous chapter. We used post-gastric lavage and then ID'd to the lowest taxonomic level and got percent composition by weight. And so my first specific objective is to assess fine scale small bass and walleye diet overlap. And so we did this for Lake Oahe. And so we used the simplified Morris-Cetus index, which used percent composition by weight of 2 species and ultimately gave you a value of 0 to 1, which 0 represented no overlap and 1 represented complete overlap. And then we divide it in kind of thirds to justify low, moderate, and high. And so looking at the results, so now we're going to be looking on 2019 with small bass on the top panel, walleye on the bottom panel, and the x-axis is going to be month, and the y-axis will be proportion by weight. And so what's going to show in a second is a 100% stacked bar chart. So it's going to go all the way up to the 1.0. However, it's going to be divided by different colors and textures that will represent the proportion of that specific prey that made up that average diet for that month. And then those floating numbers right there are just the number of diets that were used for that average for that month. And so looking at smallmouth bass, fish made up 52% of their diets, whereas inverts made up 47% of their diets. And the missing percentage is just going to be unidentifiable items. Whereas for walleye, they had a higher percentage of fish within their diets at 57%, and inverts were 43%. So smallmouth bass comparatively had more inverts make up their diets compared to walleye, and walleye had more fish. So now looking at the actual diet overlap for each month, we found that May, July, and September were moderate overlap, whereas June was low. But I want to now kind of focus and point out some trends that we noticed within the actual diets. And so we can see right off the bat, if you look at the dark blue on the walleye, that is Ephemeroptera, which is mayfly. It's an invertebrate. Whereas smallmouth bass, a large chunk of their diet is that dark green, which is Hemiptera, which is a back swimmer. It's also an invert. So what we noticed is that whenever diet was slightly higher, such as September was moderate, but it's 0.64, that was when smallmouth bass tend to have a more piscivorous diet and focus more on fish. Whereas whenever walleye focused on inverts, they were using a different invert, which potentially allowed them to not have such high overlap. And now looking at 2021, smallmouth bass now had 41% of their diets be fish, whereas inverts were 58%. And then walleye fish were 49% and inverts were 46%. So now this year, smallmouth bass had a larger percent of inverts within their diets. However, walleye still had fish being the higher percentage of their diets. And so looking at the overlap for this year, we found moderate overlap for May, June, and July. However, September was now high. And so we still see that similar trend that walleye for their invert preference, based on what we've collected, was Ephemeroptera, and they hardly ate a lot of Hemiptera, whereas Hemiptera made up a large proportion of the diets for smallmouth bass. And so again, when walleye did feed on inverts, they were kind of focusing on different inverts, whereas when smallmouth bass went more piscivorous, such as in September, there was overlap. And then it's worth also noting that during September is the first time that gizzard shad appeared in their diets, and it made up a large proportion of both of their diets, as well as other fish species. And so once again, when smallmouth bass kind of went piscivorous is when they started going to higher overlap. And so going further into the fall to see if that trend continued would be worth noting in the future. So moving on to my next question, I wanted to compare smallmouth bass and walleye isotopic niche overlap in systems with varying prey assemblages. And so to collect our isotope samples, we got adult smallmouth bass and walleye from GFP annual lake surveys, and we would collect total length in millimeters, and then we would get dorsal muscle tissue directly below the second dorsal fin, which is just that red circle on the walleye pictured there. And we also collected prey items, and we chose them based on diet contents that we collected, as well as annual seine survey data to see if it was practical for us to acquire them. And so we would take these samples back and oven or freeze dry them, homogenize them to a fine powder, and we would weigh them on a microbalance and pack them into these tin capsules, which we then sent to the Iowa State Stable Isotope Lab, where they have a mass spectrometer that would give us isotope values that are expressed in delta notation and defined as parts per thousand. And the two isotopes we're going to be focused on today is delta 15N, which is nitrogen, and delta 13C, which is carbon. And for the remainder of the talk, I'm just going to refer to them as nitrogen and carbon, so I don't have to keep saying delta. And so there's certain aspects of these from an isotopic biplot perspective that we're able to learn based on the values that you get. And so starting with nitrogen, it is known that there's a trophic level of basically every 3.4 parts per thousand for nitrogen. So the more depleted or the lower the nitrogen level, the lower the trophic position, whereas the more enriched, the higher the trophic position. And as known, think of it kind of from a food web perspective, the top predator are typically going to have a higher nitrogen value. Whereas carbon tells us more about the resources they're acquiring energy from. And from an aquatic standpoint, the more depleted, well, before I mention, I just want to mention that carbon is negative. And so the more depleted, so the more negative carbon is, it's known that it's more from pelagic resources. So an example here would be walleye consuming a pelagic prey, fish such as cisco that consumes zooplankton. Whereas the more enriched or the less negative it is, the more littoral resources they're potentially acquiring energy from. And so an example for that is smallmouth bass consumes a white sucker that has been feeding on benthic algae that's right there near shore. And for those who aren't familiar with pelagic and littoral, pelagic is your open water, whereas littoral is kind of your near shore where light still hits the bottom. Now, based on where our values end up, we can analyze them and know certain things about them. First, you can look at kind of where they end up in the isotopic space. And then we can also look at standard ellipse areas, which is the standard deviation in two-dimensional space of a bivariate. So the bivariate is just nitrogen and carbon, and they have associated air that creates this ellipses. However, something that we can look at is looking at the isotopic niche overlap of these ellipses. And so that's what we are going to focus on for this objective. And so we use our package stable isotope Bayesian ellipses in R, or CYBER, and within that package we use Bayesian overlap, which essentially gives us a value of 0 to 1, which was similar to the simplified Morisida's index for the diet stuff. And then we did a corrected standard ellipse area, which would limit bias caused by smaller sample sizes, less than 30. And so essentially it takes 40% of the spread of the data and is commonly referred to as the core isotopic niche area. And so we're going to start off looking at just comparisons of Lake Walleye from 2019 to 2021. And so here we have our isotopic biplots with Y being nitrogen, X being carbon, and then we have potential prey species as well as small bass and walleye. And so small bass are going to be the red ellipses, and walleye will be the gold. And so focusing on nitrogen first, we can see that small bass nitrogen was lower than walleye within both years. And so something that this potentially can tell us is where they are from a trophic level position, but also where they're feeding. So if they're acquiring energy from inverts, which would complement our diet study where we saw that they preferred eating a large percentage of inverts, well, then their isotope values will reflect that they're consuming potentially inverts and being lower from a nitrogen standpoint. Whereas if they have a more piscivorous diet, typically it's known that they'll have a higher nitrogen value. Whereas looking at carbon, as I mentioned, depending if it's more depleted or enriched, it can tell us potential areas of where the resource they're utilizing. And we noticed from compared to 2019 to 2021, the ellipse areas were much wider, potentially from a carbon standpoint, potentially implying that they're utilizing several different type of prey resources to acquire energy from. And as opposed to Lake Walleye from 2019 was kind of a closer ellipse from a carbon standpoint. And so looking at the overlap between the two, there was 0 core niche overlap in 2019, whereas there was low core niche overlap in 2021. And so essentially analyzing a competitive standpoint with them having low overlap, it doesn't appear that the potential competition is much of a concern within Lake Oahe. However, changes in where they're feeding may be a result. But again, this is just observation based on the lips areas and not directly the calls. We can't make that claim. And so looking at Lake Sharp, we saw in 2019 that once again, nitrogen were lower than walleye. Nitrogen for small bass were lower than walleye. However, in 2021, there was some overlap between the two, which again could be in relation to kind of where they're feeding. Whereas the difference compared to Lake Sharp, which just a reminder, this is a more shallower lake that has abundant gizzard shad annually and so less cyclical prey assemblage. We're seeing that they're acquiring energy from similar prey. So that ellipse area is now not as wide as we saw back in 2021 for Oahe. And so a thing that you can do with these is you can have potential prey that they're acquiring energy from based on how they align with the carbon, but you can't make the claim that it's definitely that because we could be missing certain prey items that could also be on here that could have similar carbon values. However, based from what we have here, it looks and appears that in 2019, gizzard shad and yellow perch made up potentially where they're acquiring energy, whereas yellow perch and H0 small bass were potentials for 2021. Now looking at the overlap, once again, similar to Oahe, in 2019 there was zero corniche overlap, whereas there was low corniche overlap in 2021. So again, kind of implying that potential competition isn't as much a concern because the resources they're utilizing in trophic position level isn't as affected. And so our last specific objective was to assess historical changes in walleye isotope signatures in a system with changes in annual small bass abundance. And so to gather historical carbon and nitrogen, we obtained those from previous literature. So we got 2001 and 2002 from Davis 2004, 2007, 8, and 9 from Finsel 2011, and then we personally collected 2019 and 2021. However, another part that can affect carbon and nitrogen when you're looking at just a single species is the size class. A smaller walleye may not be utilizing the same resources such as a larger walleye. Therefore, all of our carbon and nitrogen values we group by size class using Gablehouse Jr. in 1984, in which we had stock, quality, and preferred as our 3 size classes. And lastly, we wanted to see if changes in carbon and nitrogen were related to relative abundance of small bass. And so we got that for 2001 to 2009 from South Dakota Game Fish and Park annual reports. And so we use CYBER once again to have these ellipse areas and to analyze overlap. However, now the different colors will be different years. And so first off, we see that in 2001 and 2002, there was high corniche overlap. And so that's the red and black ellipses. Whereas now there was moderate to high corniche overlap between 2007 through 2009, which is your green, pink, and light blue. And so potentially what this is reflecting is where they're acquiring energy from and the resources they're using. It appears that in 2001 and 2002, it was more pelagic resources they could have been acquiring energy from, whereas carbon and nitrogen were more enriched in 2007 to 2009, which may reflect, again, potentially littoral resources that they're turning to and more of a piscivorous diet because it's more enriched in nitrogen. And then in 2019, so our data that we collected, it appears that it shifted back in 2019 to 2001 and 2002 levels, whereas in 2021, it became more enriched from a nitrogen standpoint. And so one thing I want to mention is that, again, these are observations, but we can't for sure say this is the reasoning behind it. But one thing that's worth noting is that in 2001 and 2002 in Lake Oahe, the rainbow smelt had a collapse within the system, and potentially some of these changes and reflections may be due to that occurrence that happened. However, that again just shows us from an isotopic space where they're lining up, but we wanted to assess changes in carbon and nitrogen specifically. So we did so using general linear models, looking at effective year, size class, and their interaction effects on carbon and nitrogen. And so we used our package EM means for estimated marginal means for our models and function pairs for pairwise comparisons. So here we're going to have the isotope values on the Y and the X is the year. And then we have the blue square is going to be stock The orange circle is quality, and then the red triangle is going to be our preferred size class. So starting with carbon, there was no size class year interaction effect or size class effect. There was only a year effect, and so these letters that are floating are denoting similarities between the two. And for example, we can see 2001 and 2002 were similar, which makes sense because there was such high overlap in the previous slide. However, looking at nitrogen, there was an interaction effect, so now those letters get a bit crazier now because you're looking at a size class within a year compared to every other size class in every other year. However, to kind of break it down for you guys, the thing that we noticed was that walleye nitrogen increased consistently with larger walleye size classes, but it was never more than one trophic level generally. And so this makes sense, and this is what the literature shows, that a larger individual will typically have a higher nitrogen level. So it makes sense that throughout size classes, as they increase, that would increase as well. However, one thing worth noting that we did observe was that in 2019 and 2021, this was the first time that the size classes all were similar based on the error associated. There was overlap between all of them. So that begs the question on why and potentially what caused them to suddenly have all these size classes occupying similar trophic positions. And so that's something we can't assess right here, but a question for the future to potentially address. And so then lastly, we wanted to determine if carbon and nitrogen were related to annual changes in smallmouth bass relative abundance, more specifically if higher smallmouth bass abundance would affect walleye's position in the food web. And so we ran linear regressions from 2001 to 2009. However, walleye carbon and nitrogen were not related to smallmouth bass relative abundance. However, it is worth noting that we only had 5 values to work with, so having a larger data set may potentially result in a different outcome. And so similar to the predation chapter, I'm now going to do an overlap / discussion summary of the diet overlap. We'll starting with the diet overlap. So walleye primarily consume fishes, whereas smallmouth bass had a higher reliance on invertebrates, with Ephemeroptera being walleye's potential preference and Hemiptera being smallmouth bass potential preference. And so that whenever walleye were feeding on inverts, potentially kept overlap to be low. However, when smallmouth bass switched to more of a piscivorous diet, it appears that that had an effect on their overlap. But overall, smallmouth bass and walleye exhibited moderate diet overlap that varied temporally, with June exhibiting low and September exhibiting high. So therefore, sampling further into the fall would be beneficial to look at whether high overlap continues, especially since we saw that in 2021, we had similar to Woolner et al. 2010, the high overlap coincided with a high proportion of gizzard shad within the diets. So again, it would warrant sampling more into the fall to see whether whenever they occurred with high diet overlap, if that was also still related with gizzard shad being a large proportion of their diets. Now looking at isotopic and niche overlap between the two systems, both had no isotopic core niche overlap in 2019 and low in 2021. However, the difference between the two systems was that Lake Sharp acquired energy from similar resources, whereas there's variability between 2019 and 2021 for Lake Oahe, which begs the question of whether that has to do with prey availability or changes in where they're acquiring prey resources. But ultimately, from a competition standpoint, appears because the overlap was low, it's not as much of a concern. And then looking at the historical changes in Lake Oahe walleye, again, nitrogen and carbon values fluctuated, potentially reflecting shifts in where they're acquiring prey energy from. However, that again is just based on observation of where it ended up from an isotopic space standpoint. However, what we did find was that size classes consistently acquired energy from similar pathways within a year, and walleye nitrogen values tended to consistently increase with large walleye size classes. And then lastly, walleye nitrogen and carbon changes were not related to smallmouth bass relative abundance. And so with that, I'll leave off with just some final big broad takeaways for each chapter. And so the first one for my chapter evaluating smallmouth bass predation on stocked walleye. We found that smallmouth bass consume Azra walleye frequently within a few days post-stocking, but overall they consume a small percent of stocked walleye compared to the literature. Whereas for chapter 3, we assess potential competition between smallmouth bass and walleye. And while smallmouth bass and walleye can occasionally predate on similar prey, the ultimate predation resources suggesting declines in either species is unlikely due to competitive interactions. And so with that, I want to thank you guys for coming, both virtually and in person, and I will take any questions that you may have.
We have lots of time for questions. Talking to the mic again. Lots of time for questions, and so I'll try to monitor the chat for hands and questions there too. And then Kyle, if you could repeat questions from the room so
Yep.
folks virtually can hear you.
Eric.
So if your walleye population is decreasing enough from competition with smallmouth bass or why are the walleye?
Okay. So the question was, if small bass aren't affecting walleye and potentially causing their declines, what are some possible reasons for their declines? And that kind of goes back to my very first slide, potentially unsuitable spawning habitat and low recruitment, as well as high anger harvest could potentially be causes for those declines. But again, there could be other aspects that aren't on our radar that could potentially be causing declines as well. Jordan.
So with the management recommendation, are there any negative effects on?
And so the question is potential effects of delaying stocking. And I would say that there might actually be more benefits to delaying stocking than negatives. One is the more you delay, the larger the individual, and there has been studies showing that the larger that you stock, the less of a risk of predation. However, a con to it is from a hatchery standpoint, they can only hold the walleye for so long, and sometimes there's not space and they have to get them out when they can get them out. So that's why delaying stocking may not always be feasible. Any questions in the chat? Dr. Morris?
South Dakota fishing game, do they have the capacity to have, hold on to the walleye longer like Iowa DNR does, or they, like 30 days?
So the question was if the GFP DNR have the capability of holding longer, and I guess I would answer that I'm not sure, but from my one experience out there, it seemed like there was a very limited time frame on when they could stock, and so it seems like from a capacity standpoint, they can only hold so much too.
So they're harvesting their juveniles out of ponds and going right to
I believe so, yep. Any other questions? So the question was, what was the strangest thing I've pulled out of a stomach? I don't know if it's necessarily strange, but a lot more lures than you may have expected. I know we did also, in 2019, I didn't get to personally see, but there was, I believe, a mouse in the diet and a frog as well. And so those are probably the weirdest, but again, not too weird, but just kind of cool to see.
All right, we have one other question in the chat. So Mark Finsel asked, using boat electroshocking, we're only able to sample near shore bass and walleye. Would you expect to see the same diet patterns for offshore predators? If you could repeat that, or just tell them that there was a question from Mark.
Okay. And so the question I'm going to be answering is the question in the chat from Mark. And so I guess to answer that, I would assume there may be some differences in the prey that they're predating on because of the area that they're staying in. However, I think from our stocking standpoint, we're stocking right near shore. So I don't think there would be like higher predation on stocked walleye deeper because those walleye are typically hanging out in the littoral when we stocked them originally. But there definitely could be changes based on where they're occupying space, whether it's in the littoral or pelagic, because the prey assemblages are different within those two areas of the lake. Any final questions?
Mark has another one for you. Can you see him or you want me to read it off?
Can you read it? Because
Yep.
Yeah.
That's fine. What do you attribute the reduced age-zero walleye consumption in 2021 compared to 2019, despite about twice as many age-zero walleye being stocked?
So again, I'm answering the question from Mark in the chat, and that's a good question. There was one observation that we saw, but it's a bit speculative. But we saw our lowest smallmouth bass predation within a single bay, and a bay that was stocked with Chinook salmon. And so prior to stocking walleye, there was Chinook salmon that were stocked within this bay, and we found a lot of them in the diets. And it happened to be that that specific bay had the lowest percentage of walleye consumed. So again, it's a bit speculative and could be occurrence, but potentially that may have had an effect on Spring Creek 2021 being a lower.
So coming from a novice fishery, why do largemouth?
So the question was, why do largemouth bass consume more stock walleye than smallmouth bass? And I guess I don't know if we can make that claim just because there's a lot more literature out there for largemouth bass predation. However, a smallmouth bass, it's lacking. And so what we found, yeah, it was a lower percentage, but that can vary within systems in different areas. So there's a potentially right now from what we saw that it was less, but ultimately, I don't know if we can make that claim just yet. We would need more supporting evidence for that.
Once, twice. All right, well, let's give Kyle another round of applause.
Thank you.
And take a 5-ish minute break and